session, we are going to discuss about gametogenesis. Gametogenesis is defined as formation of haploid gametes from the diploid germ cells in the gonads. So now first we should understand what exactly gametes means. The gametes are nothing but the sperm and the ovum. The gametes are nothing but the sperm as well as the ovum. Sperm and ovum, they are called as gametes. So these gametes are haploid in nature. Haploid means N. That is nothing but 23. They are having 23 chromosomes. So how these sperms and ovum are produced from a diploid germ cells. So the germ cells are diploid in nature. So germ cells are diploid in nature. Diploid means they are having 46 chromosomes. From this germ cells, the sperms and ovum are produced in gonads. So gonads means in case of male, the gonad is testis. In case of female, gonad is nothing but ovary. So we are going to understand in this session how sperms and ovum are produced in testis and ovary. So gametogenesis in human beings is of two types, spermatogenesis and oogenesis. So coming to spermatogenesis, so we know that spermatogenesis is nothing but formation of sperms. The sperm formation will take place in testis, especially in male. And oogenesis is nothing but formation of ovum. It takes place in the female ovary. So now first we are going to study about spermatogenesis. The process of formation of sperms in testis is called spermatogenesis. So sperms or spermatozoa, both are one and the same. The process of formation of sperms, that is the production of sperms in testis, is called spermatogenesis. So this spermatogenesis will take place in two phases. One is formation of spermatids, the another one is spermiogenesis. So now, as we have already discussed about the testis, so TS of testis shows this structure where a testis consists of seminiferous tubule. It consists of seminiferous tubules. So these are seminiferous tubules. So this is a place where exactly the spermatogenesis, that is the formation of sperms, take place. So once the sperms are produced in this seminiferous tubule, once it is produced, after the production, the sperms will enter into retate testis. From retate testis, it enters into the vasa efferentia. From vasa efferentia, it enters into epididymis. Then it enters into vasa deferens. So that is how the main region where the sperms develops is nothing but the seminiferous tubule. So we have studied that in the seminiferous tubule, TS of seminiferous tubule, if you find the TS of seminiferous tubule, we have observed that it consists of germinal epithelium cells and Sertoli cells. So these are the germinal epithelium cells. So these are called as germinal epithelium cells. Germinal epithelium cells. So in between we can see a pyramid-like structure which are called as Sertoli cells. These are called as Sertoli cells. So here, as we have already discussed, the germinal epithelium cells, these germinal epithelium cells will undergo a further mitosis and meiosis division, which results in formation of sperms. So these germinal epithelium cells, they undergo mitosis division and meiosis division. Later on, it is going to produce the sperms. So we are going to study how these germ cells will produce the sperms. So now we can tell that the sperm production of sperm, it takes place in two steps, that is formation of spermatids as well as spermiogenesis. Now coming to formation of spermatids, first we are going to study how the spermatids are formed. It takes place in three phases. So the first one is multiplication phase, second one is growth phase, and third one is maturation phase. So formation of spermatids will take place in three phases, that is multiplication phase, growth phase as well as maturation phase. So first we are going to discuss about multiplication phase. The name multiplication phase itself indicates that the cells are multiplying. So here a diploid primary germ cell will undergo repeated mitotic divisions to form a number of diploid spermatogonia. As I have told you, in the seminiferous tubule, there are germ cells and Sertoli cells, but I am talking only about germ cells now. So these are the germ cells. So these germ cells, they undergo mitosis division. They undergo repeated mitosis division, and then, so you know very well, one cell will undergo a mitosis division which gives rise to two cells which are diploid in nature. As throughout our body we are having diploid number of chromosomes, that is 46 chromosomes we have. In the same way, in the germinal epithelium also, initially it will be having 46 chromosomes. When it undergoes mitosis division, it also produces the cells with 46 chromosomes. So now, of this 46 chromosomes, you can find here the germinal epithelium cell is present with the 46 chromosome. One epithelial cell we have taken here. This will undergo a mitosis division. So when it undergoes mitosis division, it produces two cells which is of 46 chromosomes. Out of these two cells, again it undergoes mitosis division which results in formation of four cells. So from here, as it is undergoing mitosis division, this phase is called as multiplication phase. So as the germ cell is undergoing mitosis division, a complete mitosis division, we are calling it as a multiplication phase. Once the multiplication phase is over, each single cell is called as spermatogonia. Each single cell is called as spermatogonia. Now, in this cells, we can differentiate the spermatogonia. Now we are having, for example, we are having here 4 spermatogonia. Of this 4 spermatogonia, some of them are considered as type A spermatogonia. So imagine that these are considered as type A. These are considered as type A spermatogonia. Type A spermatogonia. And this is, imagine the single one we are showing, it is considered as type B spermatogonia. Type B spermatogonia. Type A spermatogonia. So these cells, now here imagine three cells. Imagine that these cells, those are having type A spermatogonia, they do not enter into growth phase. They do not enter into growth phase. But again they stay like a spermatogonia. Again they undergo repeated mitosis divisions to produce again spermatogonia. Whereas type 2 spermatogonia will enter into the second phase, which is called as growth phase. Which is called as growth phase. So I think it is clear now. In the spermatogonia, whatever spermatogonia is present, it is of two types. Type A spermatogonia and type B spermatogonia. Type A spermatogonia functions as a stem cell. Stem cell means it undergoes multiplication where it is going to produce again the spermatogonia and produce more spermatogonias. Whereas the type B spermatogonia again divide and it is going to produce 4 spermatogonial generations which later on enter into the growth phase. So next we can find what happens to this spermatogonia. Spermatogonia type B, spermatogonia will enter into growth phase. In the growth phase, the spermatogonia will grow and become almost double in size. So it becomes bigger in size due to the accumulation of cytoplasm as well as replication of DNA. And it is called as primary spermatocyte. So as you can find here, this is a growth phase. So in the growth phase, the spermatogonia has increased in its size. It is increased in its size due to accumulation of cytoplasm as well as replication of DNA. And this is called as primary spermatocyte. So two steps over, that is multiplication phase, growth phase. Next phase is maturation phase. So coming to this maturation phase, we can find that a primary spermatocyte. So this is a primary spermatocyte. This primary spermatocyte will undergo meiosis 1. We know meiosis cell division, it occurs meiosis 1 as well as meiosis 2. In the meiosis 1, the primary spermatocytes, once it enters into meiosis 1, it gives rise to two cells and they are called as secondary spermatocyte. Please concentrate on the chromosomal number. Because it is a meiosis 1, the chromosome number has been reduced here, which is showing 23 chromosomes. So meiosis division is a reduction division where the number of chromosomes will become half. So now secondary spermatocyte will again undergo meiosis 2 to produce 4 spermatids. So here you can find each and every secondary spermatocyte. You can find here two secondary spermatocytes has been formed. Two secondary spermatocytes has been formed. Each secondary spermatocyte will produce spermatids, two spermatids. So at last, after meiosis 2, we are going to get 4 spermatids are formed. 4 spermatids are formed. So they are haploid in nature. So these spermatids, they are not matured. So there is a transformation of spermatids into spermatozoa, that is sperms, called spermiogenesis. So the second event is spermiogenesis. So we can find here in the spermiogenesis, the spermatids undergo metamorphosis to convert into sperms. This process is called as spermiogenesis, or it is called as spermatilliosis. So now the spermatid, which is circular or oval in shape, it changes into the shape like a sperm with a flagella which can move. So spermatid is immature, oval in shape as well as non-motile. It is converted into motile mature sperms. The process of transformation of spermatids to sperms is called spermiogenesis. Very, very important. Now, after this spermiogenesis, we can find 4 sperms are produced. Please remember that one spermatogonium, one spermatogonium will produce spermatogonia. One spermatogonia will produce 4 sperms. Will produce 4 sperms. So once the sperms are produced, you know that all these events are taking place inside seminiferous tubule. So these were the germinal epithelium cells. It has undergone the mitosis division. It has undergone the mitosis division. It produced spermatogonia. So these are spermatogonia cells. These spermatogonia, they grown big in size to form primary spermatocytes. It has given rise to 2 secondary spermatocytes. So this secondary spermatocytes gave rise to spermatids. So you got 4 spermatids. The spermatids will later produce sperms. So these sperms will bind to the Sertoli cell. It will bind to the Sertoli cell. So you can see here, after spermiogenesis, the sperm head becomes embedded into the Sertoli cells. Once the spermatogenesis, that is formation of sperms, happened, the sperms will bind to the Sertoli cells, and then these sperms will be released from seminiferous tubule by the process called spermiation. So later on, these sperms will be released from seminiferous tubule, which is called as spermiation. So in this, you have studied 3 definitions. So one is spermatogenesis. Spermatogenesis means formation of sperms. So already the definition I have already given. Spermatogenesis means formation of sperms. So second definition here is spermiogenesis. So second one what you have learnt here is spermiogenesis. Spermiogenesis means the transformation of spermatids into sperms. The transformation of spermatids into sperms. And the third one is spermiation. So you should be more careful with these 3 definitions. Spermiation. So spermiation means it is a releasing of sperms from the Sertoli cells. Releasing of sperms from the Sertoli cells is called spermiation. So next we are going to study about spermiogenesis. As we have discussed there how the non-motile, how the non-motile circular spermatid will transform into sperms, how the spermatid has transferred into sperm. That complete process we are going to study here called spermiogenesis. So during spermiogenesis, the changes taking place are, so here we can observe that the spermatid, so this is a spermatid. This spermatid is having this nucleus is present, as well as the cell organelles like Golgi apparatus, mitochondria and centriole, cytoplasm as well. So all these cell organelles present inside the spermatids, how it is going to change, we are going to study here. So first of all, the nucleus. So the nucleus will start undergoing condensation. So condensation means it is becoming packed tightly, closer. All the contents present in the nucleus is becoming condensed, coming close, tightly packed. And while undergoing this condensation, it loses messenger RNA and acidic protein. The protein and the messenger RNA present in the spermatids will be lost. That we can say in the spermatid, DNA and basic protein will be present. So in the nucleus, we can tell that it consists of only DNA. Now, this nucleus will start undergoing condensation. It condenses in such a way that it moves anterior. You can see the last slide here. This part is a nucleus where the nucleus has moved to the tip region, anterior region of the sperm. As well as we can say it has become oval in shape. The shape it has become oval and it has become flat. So these are the changes it has taken place in the nucleus. Next we can see about Golgi bodies. The Golgi bodies which is present in the spermatids. It has occurred many changes. You can find here how the Golgi bodies is becoming cap-like structure, and here you can see clearly the yellowish shade here. So this is called as acrosomal cap. This acrosomal cap is nothing but the Golgi body. The Golgi bodies has been changed its formation in a cap-like structure which is called as acrosome, and this acrosome will have sperm lysins. It consists of enzymes. So Golgi bodies will be occupied with a sperm lysins. It is nothing but the enzymes. So acrosome will have enzymes which has a capability to digest all the membranes of the egg. So once the sperm enters and reaches the egg will have many membranes. All those membranes will dissolve due to this enzymes. Next we are going to study about centrioles. So centrioles present here, these centrioles will divide and it is going to form two centrioles. One will be present as a axial centriole and another one is called as distal centriole. So this distal centriole will form the axial filament. We can find here this is the axial filament. We will study what exactly the functions of this axial filament and the centrioles in the structure of sperm. So this is an axial filament which is formed by a distal centriole. So next we can find mitochondria. Mitochondria changes into spiral and spring-like structure and it collects around the upper part of axial filament. You can find here the mitochondria. The mitochondria has the different stages you can find here. The mitochondria is changing slightly, and you can find here these are the mitochondria. So it is become spring-like structure. Clearly you can find here. So it has become a spiral and spring-like structure. So next we can say that the cytoplasm present in the spermatid is lost. So there will be a slight amount of cytoplasm will be present in the tail region of the sperm. Cytoplasm in the head part is completely nil. There will be a little thin part of cytoplasm and it will be present only in the tail part. Hence, we can tell that the sperm will contribute only genetic material. It will contribute only genetic material. So, the complete process of spermatogenesis, it takes for 64 days. And then we can tell that human male manufactures 10 to the power of 9 to 10 to the power of 10 sperms per day. So in the next session, I am going to discuss about the structure of sperm.